Terrific. So thank you very much, Kedar. That was really well done. So now I'm going to present Albert's slides. Are we okay for 5 minutes? Okay. So in addition to the cell-based models, Kedar also mentioned that we can use animals to study. So we obviously try to minimize animal use as far as possible, but there are certain things that you cannot do without using animals in research. So this is all really Albert's work, but I'm presenting on his behalf. So Albert and I, we're working together as part of this EpiGen Rare consortium that I talked about, where we're trying to use mouse models for Kabuki syndrome to understand the disease and to find treatments for Kabuki syndrome. And we're taking slightly different approach in that we've talked about Kabuki-like conditions. So if patients have Kabuki-like features, you think that at a cellular level as well, there will be some overlap. So the mechanisms will overlap as well. And if you can understand what's overlapping mechanism biologically, then you might be able to use the same treatments for multiple disorders. So if you go on to do a clinical trial, if you say, I'm going to develop a drug that's only going to be useful for 100 patients, probably that's going to be a difficult challenge to get funding and for pharmaceutical industry, et cetera, to get interest in. But if you develop things which might be applicable for thousands of patients, then suddenly there is going to be more interest as well. So there's a biological reason, there's pragmatic reason, there's clinical reason, there's scientific reason, and there's of course, I said I'll keep on mentioning those pound signs again and again. So Albert is wonderful, and together with him, together with all of us, what he's leading on is to answer that question, that hypothesis really, as to if studying similar conditions together might help us to understand all of those conditions together, and then if similar treatments might work for. It says both conditions there because we initially, as a proof of principle, we are taking two conditions in this project. So Albert is a mouse biologist. He's an expert in using mice to study diseases. So we're trying to use a mouse model for Kabuki syndrome, and there's a similar condition called CHARD syndrome, which probably some of you might be aware of. But suffice to say that there are definite clinical overlaps between the two conditions. And what he's doing is studying the brains of these mice. He's using a special MRI scanner. So these are MRI scanners for mice brains. They do exist. And he's trying to understand what is the difference between the control mice, Kabuki mice, and CHARD syndrome mice. And then, in addition to the structure, Kedar talked about gene expression, the RNA studies. So similar RNA studies can also be done in mouse brains. And he's also trying to study the DNA structure in these mice as well. So right now, the first part of the study has been completed, but the second part is still going on. And if once we are able to complete that, then what we can do is we can give certain drugs to these mice and see if those drugs can revert the brain changes, bring them closer to the control mice rather than the differences that you see in Kabuki and CHARD mice. So that's really exciting, but that will take another couple of years for us to get there. So that brings us back to some of the work which Sarah and Kedar are doing. So we've talked about up to here. This is the last bit which is remaining. I don't really have any slides for this because there are no clinical trials, right? So there's not much to talk about. Can I maybe just complete the last bit? I might go 5 minutes over. Okay. Kedar, better you do it.
So again, so my next avenue in my PhD will be to develop to find out a drug for Kabuki syndrome, and for that I'll be doing a drug screening. So to give a background, there has been some work in the past decade about developing a drug for Kabuki syndrome, and these studies have been mostly in mouse models or zebrafish models. But what I will do is I will do a drug screening using my model system, that's the stem cells. So I'll explain how the pipeline or the procedure that we are going to follow. We'll take stem cells, put them into a multi-well plate, then try different big panel of drugs, and then I'll do some analysis with those drugs to see how the cells are functioning in response to that drug. And we'll find some drugs that might be working, that might be the hits, proper hits. Some drugs maybe we could find out as partial working, and some drugs maybe not work, and that we can just get rid of that and continue our further analysis with those drugs that are the best suited for Kabuki syndrome. So that's the overall long-term goal in our work, and we hope to find out some therapy for Kabuki syndrome soon. That's it.
Thank you. So Kedar gets 2 claps. Very good. So this is again Albert's slides. We are going to listen from hands, so I'll skip over this. So this is just to say that people have already used mice models to show that treating these mice with drug can recover some of the phenotype or some of the problems that these mice may have. We are also, as part of the Chrome Rare project, which Kedar is, we're collaborating with a French group where we're going to study the zebrafish model for Kabuki syndrome. And rather than mice, in zebrafish you can test higher number of drugs, because mice are really, they take long to develop and they're quite more expensive, whereas zebrafish, you can test several hundred drugs in one experiment. So that really leaves me to thank a huge number of people. So all these individuals. So you heard about Sarah's work, Adam's work, Kedar's work. We didn't really talk about Evgeny's work, but hopefully next time when you come back, it's just because he's doing such complex work that I don't really understand. So, and then Tom and Aoife. So thank you, Aoife, as well. These are all the other investigators that we work with, and of course, all of this cannot be possible without all the clinicians, patients, and families. Because even the stem cell work that Kedar presented, it was all from actual patients where we got those cells. So thank you very much for everyone who's helped us with that. And Ola and Pevin. So Pevin was here for a year, and she was absolutely amazing. And now she's gone back to China. Sadly, we couldn't keep her. And Victor, we've talked about, and all the funders. So thank you very much. And we're on time almost.
Yeah.